Bismillah ar-Rahman ar-Rahim. This is a data show description for the salivary glands and the pancreas. We all know that the salivary glands are of two types, major and minor. The major produce 90% of the secretion of saliva, and they have a branching duct system. They have many ducts that open finally in one main duct. So they are compound because they have many ducts opening in one final. Compound tubulo-alveolar because the secretory part is tubulo-alveolar. And the examples, of course, we have parotid, submandibular, and sublingual. In case of the minor, they produce 10% of the salivary secretion, but they constitute 70% of the mucus present in the saliva. So the main producer for mucus in the saliva is that minor salivary glands. These salivary glands are branched tubulo-alveolar, simple branched tubulo-alveolar. What is simple? Because one duct. Branched because the secretory part is branched and it is tubulo-alveolar. So they have direct short ducts opening in the oral mucosa, directly in the oral mucosa. So they are simple branched tubulo-alveolar, not compound tubulo-alveolar. And they are present under the oral mucosa, and the examples would be the labial, the buccal, the lingual, the von Ebner, and the palatine salivary glands. As you see here, these are the major, the parotid, and the submandibular and the sublingual, and the minor are scattered under the oral mucosa, as we have said. The major glands have a general structure formed of stroma that is formed of capsule, trabeculae, and reticular formation. As regards the capsule, it is dense connective tissue of the irregular type formed of collagen fibers, fibroblast. As regards the trabeculae, the septa, they divide the gland into various lobes and lobules carrying blood vessels and the ducts. As regards the reticular formation, formed of reticular cells and reticular fibers, forming a network stained with silver. Don't forget that the connective tissue stroma contains cells. The most important to remember is the plasma cell. These plasma cells produce immunoglobulin A, together with the secretory cells of the acini. Concerning the parenchyma, it is formed of the secretory cells, either serous or mucus, and these cells will pour their secretion into the second component of the parenchyma, which is the ducts. So we have acini, secretory acini, and we have ducts in the parenchyma. We have three types of acini in the salivary glands. We have the serous acinus, mucus acinus, and the mucoserous acinus. How to differentiate between these types of acini under the microscope, يعني on the practical level and not the full theoretical level. On the practical level, we have the following. Look at the shape and position of the nuclei. In case of the serous acinus, it is a central rounded nucleus, but in case of the mucus, it is basal flattened. The mucoserous, it is mucus acinus with a cap called crescent of Guyanosi, formed of serous cells at one side of these mucus cells. Now, here we have, as regards the cells of the serous, in addition to the position of the nucleus, so actually here in the picture, it is not completely central. No problem, we will look for also another more evident feature, which is the presence of the secretory granules acidophilic at the apex of the cell. Here we don't have any granules but evacuated cytoplasm due to dissolving of the mucus during preparation of the section. So here it is rounded, this is flattened. This is, according to theoretical, it should be central, but never mind. Here, the position of the nucleus is basal. We have apical secretory acidophilic granules. Here we have vacuolated cytoplasm. So, by just looking for the shape of the nucleus and the appearance of the cytoplasm, you can differentiate between the mucus and the serous. In case of the mucoserous, this is the mucus cells, and those which are more darkly stained due to the presence of these acidophilic secretory granules. Here, these are the serous cells. Here we have the dark cells at one side forming a crescent hilal of gyanosis, and these are pale vacuolated cells with the basal flattened nucleus like this one, and like this one, and like this one. But here they are of a rounded shape. Now we can also differentiate between these two types of acini. We have, this is the serous, granules, red, nucleus is rounded, and here we can see that the nuclei are flattened and basal, and the vacuolated cytoplasm. Don't forget that we have other cell around this acini, which is the basket cell or the myoepithelial cell, whose contraction will cause evacuation of this acinus. Of course, this secretion is more thicker than that serous, so we will expect to have more myoepithelial cells here than there. By the electron microscope, the differentiation is very easy. By looking just for the cytoplasm, here we have the secretory granules appear to be electron dense, very dark at the apex of the cell. But in case of the mucus cell, we will have electron lucent, means pale, fatha, and they are of course membrane bound. These are the mucus or the glycoprotein granules, the mucus granules or mucinogen granules. So this is dark and this is light. For the mixed acinus, which is the mucoserous acinus, you can differentiate the mucus cells by the vacuolation of the cytoplasm, the position of the nucleus, and the shape. And the serous cells are more dark compared to this, and the nucleus is more or less rounded. Look at this much better picture, because we have here the secretory acidophilic granules are very evident in the serous cells, and these are the vacuolated cells of the mucus secreting characters. Don't forget the name of this demillion, which is called crescent of gyanosis, hilal gyanosis. This is formed of the serous cells. And the acinus as a whole is called mucoserous acinus. This is the dominant or the main part, mucoserous acinus. Very nice picture for the mucoserous acinus in the electron microscope. Here we have the serous cells with rounded nuclei, and we have here the secretory granules, which are electron dense. But in this major part formed of the mucus cells, they are full of electron lucent granules of the mucus. And you can suspect to have this basal flat nucleus. This is to demonstrate the position of the myoepithelial cells. Myo means muscle, epithelial, as they are modified epithelial cells or basket cells. Why they are basket? Because they surround the acinus like the basket of the basketball when it surrounds the basketball. Here we have these myoepithelial cells, and this is the acinus, and this is the lumen containing the secretion. By contraction, the secretion is squeezed out to pass through the ducts. Actually, these cells are also present not only in the acini, but it is present also around the intercalated duct. In the intercalated duct, Here, as you see, this is the intercalated, this is the acinus, and these are the cells intercalated between the secretory cells، محشورة ما بينهم هنا، فدي سموها intercalated duct. These myoepithelial cells surround the secretory acinus as well as the first part of the duct system, which is the intercalated duct. Don't forget that these myoepithelial cells are present between the base of the cell and the basement membrane. Take care, it is not outside the basement membrane, it is between the base of the cell and the basement membrane. So this is the myoepithelial cells surrounding the acinus and the intercalated duct, like an octopus or like the basket when it surrounds the basketball. As regards the duct system in the major salivary glands, we have 5 ducts. Number 1, the intercalated duct, which is present in between the secretory cells. This is the first duct. Then we have the striated or intralobular duct. And then we have in between the lobules, interlobular duct, and then we have in between the lobes, interlobar duct, and finally we have the main duct. You should remember the lining epithelium of each of these 5 ducts. According to the textbook, we have the first one, which is the intercalated duct. This intercalated duct, it is the first duct arising from the acinus. These are the intercalated ducts, and they are lined, as you see, by simple cubical epithelium. Then they join together to form the secretory duct or the intralobular duct, where the cells are lined by cuboidal. Cuboidal means higher cubical cells, longer cubical cells, as if they are apexless pyramid. So they are cuboidal or columnar, low columnar cells with the rounded central nuclei. So take care, this is the cubical and this is higher. So it is cuboidal or low columnar. Cuboidal means low columnar, but they have central rounded nuclei. And then we have after that the interlobular duct, and then interlobar, and then we have the main duct. The first duct, which is the intercalated duct, as you see here in the picture, this is the acinus. What type of acinus? It is granular, it is acidophilic, it has a rounded nucleus, so it is a serous acinus. And this is the first duct arising, so it is the intercalated duct. It is lined by cubical epithelium with the central rounded nuclei. It has acidophilic cytoplasm, and don't forget they have myoepithelial cells, these flattened or oval nuclei of the myoepithelial cells, whose contraction will help the evacuation of the contents of the serous acinus as well as the intercalated duct to higher ducts. Again, look at this intercalated cut in cross section. We have the myoepithelial cell, which is flattened, but the lining cells have rounded nuclei. So these are the rounded nuclei of the intercalated, and this is for the myoepithelial cell, either around the acinus or around the intercalated duct. Then we have the second duct is the striated or the intralobular duct or secretory duct. These are 3 names for the same duct. Here we have why it is called striated, because it shows basal striations here inside the cells. Of course, we know that it is low columnar or cuboidal, and it is acidophilic cytoplasm, but it is showing, in addition to the granules, which are the secretion, it shows basal striations. We will know now what is the cause of these basal striations and what is the significance. يعني إيه السبب وإيه الفايدة. If we look at the light microscope picture, you will see these striations very evident at the base. And look carefully, you will see some granules here in the apex. So this is the striated duct or the secretory duct as it produces secretion, or intralobular duct as it is present inside the lobule. It is lined, as we have said, by cuboidal or low columnar epithelium, but take care the nuclei are rounded and not basal oval as typical columnar cell. Here we have the acinus formed of these serous cells, and here we have these are the serous cells which are granulated, serous acinus. Here we have the granulated cytoplasm, apical acidophilic granules, and the rounded nucleus. And we have here the myoepithelial cell. Look at the plasma cell in the connective tissue. As I have said before, these plasma cells will produce immunoglobulin A in addition to the secretory cells. So it has an immunological function here. Very important to diagnose this plasma cell, which has a cartwheel appearance for its nucleus. So this is the striated duct, and look, it is most commonly, it has a larger or a bigger size than the serous acinus. Now, we will explain why these striations are present and what is the significance for that. Here again another LM picture showing the striated duct. How you diagnose this duct? By the presence of the striations, these striations at the base. The appearance of the cells, it is cuboidal or low columnar with central rounded nuclei. So this is the striated duct or the secretory duct or intralobular duct. Again, here we have the striated duct or secretory duct or intralobular duct having these striations and the acidophilic cytoplasm evident here. What is the cause of these striations? The cause of these striations, it is the basal infolding. The cell membrane is not straight, but it causes basal infolding. And inside these irregularities, we can see these mitochondria, and they are very helpful in the function of this cell, which is ion transport. So we have this interdigitation and basal infolding in the base of these cells. We have basal infolding to increase the surface area for the ion transport, and this demonstrates that this cell is performing ion transport, as we will say now. And also for the ion transport, also we need the mitochondria for the active transport energy. So we have a typical picture of ion transporting cell, which is the presence of basal infolding and the presence of the mitochondria at the base of these cells in between these infolding. Here, this is the cause of these striations. What is the value of this striation? Actually, this cell will perform what? This is the base of the cell forming basal infolding, and in between this infolding we have the mitochondria. And this, as we have said, and this will increase the surface area for the ion transport. What ion? Actually, this cell is called secretory cell because it secretes here to this is the basal membrane, and then here we have this cell. As we see here, this is the cell, and this is the base of the cell. What is the function of this cell as regards the secretion? This cell will reabsorb the sodium from the saliva, and this will make the saliva hypotonic. يعني ده اللي هيخليها hypotonic. In order to maintain that and produce this hypotonicity very rapidly, this also sodium will be pumped to the bloodstream in the blood vessels. So there is continuous uptake of this sodium and pumping it to the blood so that the saliva will be very rapidly hypotonic. Actually, it also produces another function, which is the secretion of two things: secretion of potassium, but of a little amount compared to sodium, as well as secretion of the bicarbonate. So it adds bicarbonate to the saliva as a way to obtain the optimum pH for the enzyme amylase. So that this is the secretory function of this cell, which is the secretion of potassium and bicarbonate and absorption of the sodium. And this basal enfolding help the rapid release of this sodium to the blood so that it can absorb more sodium again. Now this is the excretory ducts, which are the remaining three ducts. Type. What about the secretory ducts? The secretory ducts are the intercalated as well as the striated. We have talked about the striated duct function as regards the secretion. It secretes the potassium and bicarbonate in the saliva and absorbs the sodium. Type. What about the intercalated? Actually, the intercalated duct also secretes bicarbonate in the saliva and reabsorb chloride from the saliva secretion. But now for the excretory ducts, we have the interlobular duct, and we have also the interlobar ducts, and we have the main duct. Look here for the interlobular duct. Here it is present between the lobules. It is lined by columnar cell with the basal oval nuclei. So this is the third one. And after that, when they collect to interlobar, they are now pseudostratified columnar epithelium, which is mentioned here as large ducts. These are the interlobar ducts. Then we have the main duct, which is lined by the stratified columnar شخصيا, and at its distal end, where it opens in the oral cavity, it is transformed to stratified squamous non-keratinized epithelium opposite the upper second molar tooth. Now this is the picture of the parotid gland, which is the largest gland. We can see here the fat cells. We have here fat cells, a lot of fat cells in the gland. We have here the serous acini, formed or lined by the serous cells with their characters, and then we have the duct system in addition to the blood vessels. And this duct system and blood vessels are present, of course, in the septa or the trabeculae. If you notice here, we have all the structure or the parenchyma is formed of serous acini. Again, this is a low power for the parotid gland, formed of pure serous acini. All of these are serous acini. And then inside the lobule, this is a lobule, we have the striated duct. The striated duct with the features we have said before. And then we have in between the lobules, we have also septa that contains blood vessels as well as the interlobular ducts. Here we have the lobule, serous acini, and we have here the start of the duct as it opens inside the acini, so it is the intercalated cubicle, and then it will form the striated or the intralobular duct formed of cuboidal or low columna. Here we have also, this is the connective tissue, and this we have an artery thick wall, narrow lumen, ganglion nerve cell, and here we have also the duct, the start of the excretory ducts, which are the after the striated we have the interlobular ثم الانترلوبر ثم المين duct. As you see here, it is pseudostratified columnar. So if you are asked what is the type of this duct, it is the interlobar duct, because it is pseudo-stratified columnar. But if it is stratified columnar, it is the main duct, or non-keratinized stratified squamous, it is the final part of the main duct. Note also here that the striated duct is bigger than the serous acini. What cell you expect in this connective tissue to find? It is the plasma cell, to produce immunoglobulin A that passes also to the saliva to help in the immune against any pathogens in the oral cavity. So again, this is the serous acini, this is the secretory or striated duct or intralobular duct, and this is the excretory, the start or one of the excretory ducts, where it is lined, it is big, it is in between the lobes, and it is wide lumen, and it is lined by pseudo-stratified columnar. This is a blood vessel. This is a very nice picture demonstrating, number one, these fat cells, which are present in huge amount inside the parotid. Here we have, this is a blood capillary. And then we have, these are the serous acini, and then this is the striated duct, lined by low columnar with central rounded or cuboidal epithelium, and with acidophilic cytoplasm and of a bigger size than the serous acini. And in the connective tissue here, related to this duct, you can find the plasma cells. You know, of course, that the main duct has a name here in the parotid, Stenson's duct, and it is lined by stratified columnar epithelium that changes to non-keratinized stratified squamous as it opens in the oral cavity, opposite the second upper molar tooth. The second, this is the first, and this is the second upper molar tooth. The second gland is a submandibular gland. As you see, in the human being, 90% of the acini are serous. So the main type of the acini here are the serous, darkly stained acini. And we have 10% in the form of mucus. These are the mucus with the pale appearance of their cytoplasm. And in addition to that, we have also mixed acini, which are the mucoserous. This is the mucus cells, and this is the crescent of gynose. Don't forget that this is a connective tissue septum, and also it shows some of fat cells. This picture demonstrates the various histological features that you can mention in your exam for the submandibular gland of the human being. Here we have the major type of the acini is the serous acini, and less of the mucus as well as mucoserous. So we have here, these are mucoserous, mucus with a crescent of gynose, so these are mucoserous acini. They have also the connective tissue septa full of the fat cells. And inside the lobule here, you can see this is intralobular duct or striated duct. Also, we have here the intralobular duct. So what is present inside the lobules? It is the intralobular ducts. Then we have, after that, between the lobules, interlobular, and then between the lobes, interlobar, and finally the main duct. This is again a picture for the submandibular gland of human being, where the major type of the acini is the serous acini. We have less number of the mucoserous acini. where is the mucus cells, and these are the serous cells forming crescent of gynose. Also, we have this is the intralobular duct or striated duct, or sometimes it is called secretory duct, as it produces secretion, and nothing else here. We have just the acini, the ducts, the other less numerous type, which is the mucoserous acinus. This is also the submandibular gland, because we have the major type is the serous. Here, all of these are serous. This is mucoserous with the crescent of gynose. Here, can you see it? And also we have multiple ducts, branching system of ducts. So these glands, all the major salivary glands, are compound tubuloalveolar, as it has many ducts opening finally in one final main duct. So we have the intralobular ducts, as you see, lined by low columnar with central rounded nuclei, and in addition to the other types of ducts. Here we again have a named duct, which is the Wharton's duct, opens in the floor of the mouth at the side of the frenulum. This is the frenulum, and on other side we have the opening of the Wharton's ducts. This is the picture for the third gland, which is a sublingual gland, which is the smallest one, and it is characterized by having the predominant type is the mucus acini. These are mucus acini. In addition, they have mixed acini, but in the form of mucoserous acini. No pure serous in this gland. So we have mucus and mucoserous. The predominant type is the mucus acini with their typical vacuolated cytoplasm, basal flattened nucleus. Here you should remember that they have a thin capsule but thick septa. This gland has a thin capsule but thick connective tissue septa. Very nice picture demonstrating predominant mucus acini. Here either pure mucus or mucoserous like this. These with the cap are mucoserous acini. All of these have the crescent of gynose. And here we have pure mucus acini. And these are the mucus cells. This is a crescent of gynose. This is the dark crescent here, represent the crescent of gynose formed of serous cells. Also notice here the ducts. The duct that starts or opens immediately in the acinus, this is the intercalated duct. This duct, which is aligned by the low pyramidal or cuboidal cells with central rounded nuclei, this is the intralobular or the striated duct. Here you can see the lumen of the mucus acinus, and these are mucoserous acini, as this cap of dark cell represent the crescent of gynose, while this one is pure mucus, and this is pure mucus also with no crescent of gynose. Also again, this is a secretory or striated duct or intralobular duct. And all these pointed by the blue arrow are the crescent of gynose. Finally, you have to differentiate between the three types. This one purely serous acini with evident intralobular ducts. This one is purely, or most of them, it is a submandibular gland of the human being. It is mostly serous and some of mixed acini in the form of mucoserous acini, mucoserous with crescent of gynose, and here it is mostly mucus acini with some mixed in the form of mucoserous acini. This is the sublingual gland. So the main type here is mucus, the main type here is serous, And the main type here is serous with the presence of the mucoserous acini as a lesser extent. To summarize that, the parotid is purely serous. The submandibular of human is actually seromucous. Seromucous gland where 90% are 90% serous. وال 10% اللي فاضلين هيبقوا عبارة عن حاجتين mucus and mucoserous. In case of the sublingual, it is a mucoserous بحيث إن الغالبية mostly are mucus. هم دول الغالب. And with a mixed mucoserous with a mucoserous. But no pure serous acini in this one. Identify the type of acini in A and B. Here the acini are mucus acini and this is the serous acini. What the histological characters? You can comment on the shape of the cell, the shape and position of the nucleus, and the vacuolation of the cytoplasm. Here we have the cells to be pyramidal and with the acidophilic secretory granules in the cytoplasm and with the rounded nuclei. The type of acini is the mucoserous acini, where the green arrows point to serous cells of crescent of Guyanosi. How did you know that it is serous? Because of the presence of the granules, acidophilic granules in the cytoplasm and the rounded nuclei. And these are mucus. How did you know? By the position and shape of the nucleus and the vacuolated cytoplasm. Based on the location of the ducts D, in photos A and B, identify their type and mention their epithelial lining. As regards A, we have this is the secretory duct present inside the lobule, and it is called secretory or striated. It is lined by low columnar with central rounded or cuboidal. As regards the photo B, this duct is present in the connective tissue septa and it is lined by pseudostratified columnar, so it is the interlobar duct. The structure marked by the black star is the striated or the intralobular duct. The structure marked by the red star is the serous acinus. Mention two criteria for differentiation. The duct here has a wider lumen and a bigger size than the serous acinus. Number one. And number two, here we have striations at the base of the lining cells. But in the serous acinus, we have acidophilic secretory granules at the apex, and they have basophilic part. So these are the two criteria. You will mention the size and you will mention the staining of the cytoplasm. All of this is acidophilic and with striation, but this has an apical acidophilic granules and basal basophilic part. Identify the type of salivary glands in the following figure. Figure A. You can notice here that the serous acini are the only type present, and they have a lot of fat cells in the trabeculae, and they have well developed striated or intralobular ducts, so this is the parotid gland. The second slide, B, you can notice the presence of the mucoserous acini together with serous. We have serous acini, we have mucoserous acini, but the predominant type is the serous. We have some of the fat cells in the connective tissue. This is the submandibular gland of human. So in this picture, you have serous acini, we have mucoserous acini, the predominant is the serous, we have mucoserous acini, and we have mucus acini, number 2. This picture is for the sublingual gland because we have the mucoserous type of gland where the predominant type is the mucus acini. These are mucus acini together with the mucoserous acini. This is mucoserous because this is a crescent of gynosis here and there. And this, of course, is the intralobular duct. This picture, of course, is for the parotid, formed of pure serous acini, and we have a lot of fat cells in the connective tissue septa, and we have prominent striated or intralobular ducts inside the lobule itself. Identify the labeled structures. This is mucus cells, and this is a crescent of this is mucus cells, and this is crescent of gynosis. So this is the mucoserous acinus. Here we have also crescent of gynosis, serous demilion, and mucus cells. Here we have the serous acini, typical serous acinus with the acidophilic apical zymogen granules, serous acini, and this is the one of the duct system, which is formed of cuboidal or low columnar with rounded nuclei, central rounded nuclei. This is the striated duct inside the lobule but cut in a longitudinal section. And this is a mucoserous acinus. And this is a mucus acinus. And this is MC. It is the myoepithelial cells. Here we have a myoepithelial cell for the squeezing of the gland. Of course, this is a picture for the submandibular gland of human where the serous acini predominate. The yellow and white arrows, the yellow is for a serous acinus, the white for the striated or intralobular duct, and these of course are fat cells, and the gland is a parotid gland. Identify acini 1. 1, it is a mucus acinus, and 2, it is a mucoserous acinus. This is the crescent of gynosis in this mucus acinus. And of course, this is the sublingual gland. This is a very nice EM picture demonstrating the cells of the striated duct or intralobular duct. This is the basal membrane of the cell in the duct system of a major salivary gland. Identify the cell. It is the cells lining the striated duct. Describe the histological structural criteria. These cells are low columnar with central rounded or cuboidal epithelium, and they have basal striations and a lot of as a basal striation in the light microscope and apical acidophilic granules. By the electron microscope, they have basal infolding together with elongated mitochondria in between these folds. These features are characteristic for ion transport cell. So take care of the question if he asked about LM or EM. If he asked about EM, this is the basal striations and the elongated mitochondria in between the basal infolding. And these features are characteristic for ion transport cell.